Dear colleagues, dear academic community members of Babes-Bolyai University, it is a great pleasure to welcome you today to the today's session of Studium Generale series hosted by the STAR-UBB Institute. This is an interdisciplinary meeting space for teachers, researchers and students of the Babes-Bolyai University. Today we stand at the fascinating intersection of computational physics and medicine, exploring how network science can help us decode the complexities in human health. We are honored to have with us Dr. Dávid Deritei. It is a particularly special occasion as we welcome back a member of our own academic family. Dávid is an alumnus of the Faculty of Physics here at Babes-Bolyai University, where he completed his bachelor's degree in physics and his master's degree in computational physics. He continued his academic journey by earning his PhD in network and data science from the Central European University. Later on, his career has been marked by significant international mobility and interdisciplinary research, including positions at Pennsylvania State University in USA and Semmelweis University in Hungary. Currently, Dr. Deritei is a postdoctoral research fellow at the prestigious Channing Division of Network Medicine at Brigham and Women's Hospital, Harvard Medical School. Today's presentation, titled Complex Diseases, Solving the Genetic Murder Mystery, addresses some of the leading causes of death worldwide, such as heart disease and COPD. Before I invite Dr. Deritei to share his insight with us, I remind our audience that the presentation will be followed by a discussion session. Questions can be posted also live on the Babes-Bolyai University's YouTube channel, where this conference is being broadcast. We will address these questions at the end of the presentations. Dávid, welcome back to Cluj, and the stage is yours.
Thank you very much. Thank you for the kind introduction, and it's great to be back again. So today we are going to try to solve a murder mystery. It's a big murder mystery that involves one of the most prolific killers of our society, complex diseases. So let's maybe start with the definition. What is a complex disease? Complex diseases are conditions that emerge as a combination of multiple genetic factors, lifestyle choices, and environmental factors. Lifestyle can be diet, exercise, smoking. Environment can be pollution. We will be mostly focusing on the genetic part. So, as I said, probably in our day-to-day lives, we all have people in our lives who are suffering or have passed away due to some of these diseases. This is a screenshot from the WHO, the World Health Organization website, for the top 10 causes of death in 2021. And this is on the x-axis, we have the number of deaths in millions. On the top, we have heart disease, then third is stroke, chronic obstructive pulmonary disease, and all the way, except for COVID and tuberculosis, all of these diseases could be considered complex diseases. And even if we count COVID or tuberculosis, where we know the pathogen, the response to the infection can be very, very different due to different genetic backgrounds. So this is our murder case, and I am going to mostly focus on one of these diseases, COPD, or chronic obstructive pulmonary disease, which was in the 4th position here, responsible for almost 4 million deaths in 2021 worldwide. COPD is a disease that emerges as a consequence of smoking and aging, but this is mostly prominent in the Western world, but in countries like China or India, where there are higher rates of pollution, pollution can also create these kind of complications. And COPD has mainly two kind of symptoms. One is the tightening of the airways, which we call chronic bronchitis. It's a thickening of the airways that causes this kind of obstruction and makes breathing much more difficult. And the other is the destruction of the alveoli. The alveoli are these, I like to think of them as like these grape-shaped structures in our lungs that can expand and retract, and they are responsible for, they create this big surface, and then they are responsible for efficient oxygen exchange. And over time, with the injury caused by pollution or smoking, this alveolar structure gets destroyed. And this is what we call emphysema. It's the destruction of the alveolar structure. So I am mostly going to focus on COPD because our lab at the Channing has been historically also focusing on lung diseases, and we have a lot of biological data collected. However, all the methodology I'm going to talk about today, it can be applied and is applied to most of these complex diseases. So this will be more of a general application. So in order to better understand the problem, let's take a very simple example. We have two fellows, Steve and Bob, who are friends. They are of the same age, they are the same sex, they are neighbors, they live in a similar environment, they have similar lifestyle choices, they are about the same weight, and they smoke, both of them, about the same amount. However, by the time they reach 55, Steve is diagnosed with COPD, while Bob is not. As I said, the environment, the lifestyle, age, sex are all similar or the same. So where is the difference? And the difference must then lie in our genes, or in their genes specifically. So a little bit of a high school biology recap. Every cell in our body contains a big molecule, which we call the DNA, and this molecule contains instructions on how to build our bodies. It contains instructions on what different cells to differentiate how our organs emerge, and so on. We all start in a little embryo that has one DNA, and then our bodies emerge from that. And different sections of the DNA, you can imagine it as a long line, if you stretch it out, which has billions of little so-called nucleotides, which we usually denote with 4 letters, A, T, C, and G. And we have billions of these nucleotides repeating, they are in pairs. And on this long line of the DNA, there are different sections, which we call genes. What is a gene? It's basically just an interval on the DNA that gets transcribed into a molecule called RNA. This RNA gets out of the cell's nucleus, and it's translated into a protein. And proteins, you can imagine as little machines that do stuff. They can bind to each other, they can create these complexes that have certain functions, and this determines what the cell does, or what kind of cell it becomes. So if we go back to our friends Steve and Bob, what we can do is try to compare their DNA. And the big breakthrough, big scientific breakthrough of the early 2000s was that we were able to sequence the entire genome. So the technology became available to sequence the entire genome, which means that we can say we can measure every single letter of these billions of letters on the DNA. So what we can do is to take an entire genome sequence of Steve and an entire genome sequence of Bob and then compare them. And what you can see is that maybe this is a very, very small section of that, but most of the letters agree, like over 99% of the genome agrees, unless they would be twins, but they're not twins, so there will be differences. And when we find a difference of a single letter, like here I highlighted, that is what we call a single nucleotide polymorphism, or short is for SNP. When I say SNP, it usually refers to some sort of mutation that is different between two people. Actually, technically, we are comparing these two people to a reference genome, but it's easier to imagine that we are comparing them to each other. So we actually are going to find millions of these SNPs when we compare two genomes. However, we don't really know which of these mutations cause Steve to be susceptible to the disease and which are just maybe their eye color or their hair color. There are natural differences. There can be differences in parts of the genome that don't do anything. So how do we overcome this? And one of the ways of overcoming this is to collect a lot of Steves and a lot of Bobs. By that I mean a lot of people who are of similar lifestyle, of similar other factors, and separate them into two groups: people who develop COPD and people who do not. And now this becomes a statistical problem. And we can come up with statistical models where we are looking for mutations, we are looking for SNPs that predict the separation of these two groups. We try to average out everything else, age, ancestry, environment, and try to find those mutations that can separate these two groups. And these mutations are our first clues. And these type of studies are called genome-wide association studies, and they have been conducted for many years now, especially, for example, on COPD. So this is a study from our group from about a little bit over 10 years ago, where we tried to identify SNPs that separate large cohorts of COPD versus control genomes. On the x-axis here, we show a small section of the genome. It's not the entire genome. And every single point is a SNP. And the height of the points is actually the statistical significance of how well this SNP separates COPD from control groups. And this is on a logarithmic scale. So here, when we are thinking about p-values in general in statistics, usually we say, oh, statistically significant means p is less than 0.05. Here we are looking for p-values around 10 to the minus 6, 10 to the minus 8, because we have so many points that the significance has to be much higher. And what you see here on this figure is that many of these SNPs come in clusters. They are bunched together. That is one thing. And the other thing is that here on the bottom, I don't know how visible this is, but there's a little highlighted section. And that is actually a gene. So this section of the genome is actually coding for a gene. And the SNPs are pretty close to that, relatively speaking. Here is another gene, and the SNPs are actually on the gene. So the gene itself is mutated. So generally, when SNPs are close to a gene or are on the gene, that means that they might change how the gene normally operates, or they change something about the gene. If they are close to it, maybe they change the regulation of the gene. So now we have some candidates that we can think of like, okay, we can try to understand what happens biologically due to these mutations. So when we identify a gene like this, this is just still like a correlation. This is the statistical association. And it says nothing about the biological cause. So the next thing we can do is to try and see if actually this gene has any functional role in COPD. And this is from a study from, again, our lab from 2016, where we took this gene, HHIP, which I'm going to talk about a lot today, and we knocked it down in mice. So every gene has 2 copies. If we knock down both copies of HHIP, the mouse is dead. It doesn't develop, the lung doesn't develop. However, if we knock down only 1 copy, that is the plus minus, and we have a control mouse where the HHIP is intact, both copies, and we smoke these mouse. We expose these mice to smoke. What we saw, or my colleagues saw, that the mice that had only 1 copy of HHIP, or as we call them, they were haploinsufficient HHIP, they had a much higher likelihood of developing emphysema, which is the destruction of the alveoli, than the control mice. It wasn't a perfect split, it's still just a kind of a correlation, but this was evidence that the gene itself, where we have the mutations, had something to do with it. Now the bad news is that this is just one example of genes that have significant SNPs. Canonically, now there are about 83 genes for which we have genomic evidence that they have relevant mutations for COPD, which emphasizes the problem that this is a multigenic disease. A lot of genes play some sort of role. If you think about diseases like sickle cell anemia, the mechanism can be explained with 1 gene, but here we have a lot of genes. So over the years, we could narrow down this list of 80-something genes that on a subset that show up consistently in these studies, and for which we also have other kind of evidence, like with the mice. And so these genes will be our suspects. And today, I am going to, I'm mostly going to focus on HHIP, but I want to emphasize that we have evidence for a lot of genes, and for most of these, we don't really understand what goes wrong. For many of these genes, we don't even know what they do. This is one of the problems. The other problem is that these genes do not exist in isolation. So, as I said, the DNA sequence gets turned into an RNA, then it's turned into a protein, and the protein is floating around the big soup of the cytosol, or even goes out of the cell. But these proteins and gene products are interacting with each other. This is a self-organizing system. The DNA contains the instructions, but it doesn't have the execution power. The execution is done by the proteins. And the genes and the gene products are constantly interact with each other. They turn each other on, they turn each other off, they play off each other. And well, what is a good way of representing interactions? By a network. So this is where my expertise comes into play, because if we represent these systems with networks, we can kind of study the system-wide properties of these genes, and we don't necessarily just study them in isolation, but we study them in the context of other genes and how they interact. However, and people have realized this a long time ago, so one thing that we started to measure is what protein interacts with what other protein. So if I have one of my favorite proteins, and I can look what other proteins does it stick to, and I can draw them on a map and draw a line, and then look at the other protein and see what they interact with and draw another line. And we have 20,000 genes, which means 20,000 potential proteins, and if we draw the lines between all of them, then we get something like this. And the scientific term for this is a hairball. And hairballs are not very useful, they can be useful in certain contexts, but understanding the contextual mechanism of one gene, not necessarily. So what we have to do is to maybe zoom in a little bit and look into something like groups of genes that have some sort of shared function. And I know that Ravas Regan has given a talk a few weeks ago here, and she probably has shown something similar to this. This is actually her figure. So when we try to narrow down the problem a little bit, then we are looking for genes and gene products that have some sort of shared function. We call these maybe functional modules. And the way I like to think about the cell as sort of a hierarchical bureaucracy, where there are groups of genes that are responsible for making decisions about one specific problem. One example for this is, for example, the apoptosis. Apoptosis is a process where the cell decides to kill itself because maybe the DNA got too damaged and we don't want cancer. So apoptosis is handled by just a handful of genes that are kind of like monitoring the environment, and if there's an accumulation of signals, for example, for DNA damage, then it switches over and it starts the programs that kill the cell. So by studying these modules, these functional modules, we can maybe try to break down and reverse engineer what the role of individual genes might be. So I'm going to get back to this with a very concrete application, but I'm going to maybe step back one more time and hopefully, no, okay. I think the thing disconnected again. I'm going to talk and then maybe start the video. I want to show a video of a wound healing. However, so maybe just try to imagine if you think about COPD again. There's a sustained injury in the lung, and that causes some problem. Some people experience a destruction of the lung tissue, some people less so. So in the response to this injury, there has to be something that goes wrong. And when there's this injury due to smoke or pollution, the first layer that gets injured is the so-called epithelial layer. Now, epithelial cells are a certain type of cell that cover our whole body and cover all of our organs. Our skin is made of epithelial cells, our organs are covered of epithelial cells. They are a type of barrier, they form a type of barrier. And they are characterized by expressing genes that create proteins that make the cells stick together. They create these tight junctions, so there are certain proteins. For example, there's the gene CDH1 that encodes a protein called E-cadherin. E-cadherin climbs out from the cell and latches onto another cell, and they create this tight junction, and they create a layer. And when there is some sort of injury, miracle, this epithelial layer is the first thing that gets injured. And this epithelial layer can also heal. And this video will be of an epithelial layer grown on a plate that we injure. And this is just a simple physical injury. We can take a sharp object and just scratch it. And here you will see what happens when we scratch an epithelial layer. This is sped up. This is not real time. But what you can see, that the cells on the edge of the wound start moving almost perpendicularly to the injury, and then they just close it. So we can do it again. The other thing you can pay attention to is that the cells on the edge of the epithelium, here they are all tightly packed, they kind of detach a little bit, but not completely. And then they reform the confluent layer. So this is due to a certain program that we call the epithelial to mesenchymal transition. These sound like crazy words, but I'll try to explain them the best I can. So epithelial is a state where the cells stick together and certain proteins are getting expressed that make them stick together. Now, if something switches these genes off, then the cell can detach from the epithelium and then just go on its own, go solo. And those cells we call mesenchymal cells. When a mesenchymal cell detaches, it gains the ability to move, and it can wander off and go to other places. This is actually very important for embryonic development because that's how things grow. But also, in terms of, like, for example, cancer, this process is very important because that's how, for example, metastasis happens, that certain cells detach from the tumor and go somewhere else. Here, we see something. So epithelial to mesenchymal is just basically we turn off the genes that make these cells epithelial. We turn on genes that makes this possible, and also turn on the migration part, and then the cell becomes solo. What we saw here is something in between. So what you could see that the cells at the edge of the wound didn't completely detach. They detached a little bit. And this is what we call a partial EMT, that they loosen a little bit the junctions. They downregulate those genes a little bit because when moving, they need to be a little bit more flexible. And then when they touch again, then they can reform this epithelium. So this is on the high level. And if we think about tissue injury, this process has to play some sort of role. And this is where I start to go into hypothesis territory, because next we try to understand what is the genetic program, what is the network that controls this. And here is one way this can happen. So here is our epithelium cells sticking to each other. I create some sort of injury. These cells in the middle die. And there is a pathway called the Sonic Hedgehog pathway. Sonic Hedgehog it is actually named after the video game character because molecular biologists have a weird sense of humor. The molecule itself reminded I think people of the video game character. So Sonic Hedgehog is a so-called ligand that sits on the membrane of the cell. It's almost like a sensor. And if some sort of signal activates it, for example, a signal due to injury, the cell's dying in the middle, it starts a chemical like cascade. It's a signaling cascade. And usually a signaling cascade has some sort of gene at the bottom that gets activated. And on the bottom of Sonic Hedgehog pathway is a transcription factor called GLI1 and 2. Transcription factor sounds scary, but it's just basically a type of molecule that can climb into the nucleus and then can turn on or turn off certain genes. And so when GLI gets activated by the pathway, because Sonic Hedgehog is sensing something weird up there, GLI climbs into the nucleus and activates certain genes who are responsible for the epithelial to mesenchymal transition. So remember that when I'm getting detaching from the epithelium, that type of program. However, GLI also activates another gene, HHIP. And HHIP has a very specific role. It also climbs out of the cell onto the membrane, and it basically binds to Sonic Hedgehog. And when HHIP binds to Sonic Hedgehog, it blocks the signal. Sonic Hedgehog cannot activate anymore. So what GLI does here basically, so Hedgehog activates the signal, GLI starts the transcription, and HIP then blocks its own signal. This is a negative feedback loop. This is a negative self-regulation. And what we hypothesize here is that GLI uses this as a sort of self-moderation. So it moderates its own activation by activating HHIP, which then leads to the other genes having a moderate signal. And if these guys have a moderate signal, then the EMT also has a moderate signal, so the cells don't completely finish detaching, but move, they loosen the connection a little bit, and then they move together and they close the wound. And maybe you can probably guess where I'm going. What if HHIP is dysfunctional? Maybe due to a mutation or because we knocked it down. GLI no longer has the self-moderation, and the signal of GLI can go up as much as it wants. And then these guys can turn on, and the full epithelial to mesenchymal transition can turn on, which means that cells on the edge of the wound can completely detach from the epithelium. What does this mean? Mesenchymal cells are like rock stars who leave their band. They don't play well with others. They start to secrete signals that also turn other cells into mesenchymal cells. They don't help with the wound healing. They don't have this directionality. They don't reform. They don't join back into the epithelium unless they get like very specific signals. And they also secrete stuff that increases inflammation. And they also cause something we call fibrosis. So this is not good. This is what we call abnormal wound healing. So this means, and you don't necessarily have to imagine this like happening acutely, but if you have we can also just think about probabilities here. But if you have a sustained damage to your lung epithelium for decades by the smoke or the pollution, this abnormal wound healing can accumulate over time and can then cause the tissue to be damaged or destroyed. So this is actually now a crime scene where we have a hypothesis for one of the genes that we identified through the mutations and functional studies on what it actually what is his role in wound healing. So where did this come from? We can actually build models, network models, where we can simulate this. Why would we simulate this? Because simulations of models is much cheaper than experiments, and we can play around with this type of models. Now, don't have to get scared from this, but this part might be more interesting for physicists. And so this is a network model that we built. Every interaction here has some evidence in molecular biology. Every interaction you see here has a paper in our supplement, at least one. And this is a so-called Boolean model where every node, every variable here represents some sort of gene or gene product and is represented by a binary variable. It's 1 or 0. You can imagine it like a light switch. And the arrows pointing to every one of these nodes can change the light switch of each of these nodes. But it's not random. Every one of these has some sort of a logical function that we kind of infer from the biology of these. And then we can simulate the collective states of the whole network. And just to highlight the structure here, this purple part is the sonic hedgehog pathway. The yellow part is genes responsible for the epithelial mesenchymal system, that's that module. And this is a partial representation of the cellular migration program. And I have some inputs that correspond to some sort of damage that can activate then the certain signals. And another thing that needs to be, that's important about these models is that while all of this looks very complicated, these systems, if they are built well, have states that we call steady states, which they like. So what does that mean? Every one of these nodes is, you can imagine they are in a sort of a tug of war where certain nodes are increasing their concentrations, other proteins are decreasing their concentrations. They are all, it's a multi-dimensional tug of war where they are regulating each other. But there are certain states where everybody is happy. And these states we call steady states, and they actually match biological states. Also, we call them attractors, but let's stick with steady states. One of the steady states of this system is the epithelial state, when the cells are sticking to each other. In that state, E-cadherin, which is the glue, is turned on. Some of the microRNAs that help E-cadherin are also turned on. Everything else is turned off. And the system is happy here, and nothing will change. It's a steady state. Another steady state is the opposite. When E-cadherin is turned off, the rest of the EMT program is turned on, and then the cell, that represents the state when the cell just detached from the epithelium. That is another steady state. And we can simulate the different transitions between them. And the other thing that we can simulate is how the system responds to damage. So what happens if I turn on damage? So here is a simulation. And a very small side note, you can see here continuous concentrations. And the way we do this is just more for the physics people, that we exploit negative feedback loops. So we built into the system negative feedback loops. And when we have negative feedback loops in these systems, then it usually means some sort of oscillation. So most of the nodes oscillate. They don't stay in one when the oscillation gets activated. And if we average out a lot of these systems, because they oscillate at different rates, we can get these very nice continuous concentrations. I would say that these are like probabilistic concentrations. Okay, so, and this is a way that we overcome the binary limitation that biologists don't like, that you represent something with ones or zeros. But this overcomes that. So what do we see here? We start, this is time, and this is activation level of one of these genes. And we start in an epithelial state where everybody's happy, nothing's changing. And at 0.300, I introduce some sort of damage. I turn on the damage node. And what you have to only pay attention to a few lines here. There's a blue line here that represents glee. Because hedgehog activates, glee turns on, but it immediately goes down because it activates HIP as well, which you remember is a negative, it's an inhibitor of the pathway, and they all nicely stabilize on like 0.5. The other thing that you might want to pay attention to is this dotted line, which is E-cadherin, which is the glue, the protein that represents the glue between the cells. It goes down a little bit, but it doesn't go down completely, which represents the fact that these cells can migrate at the edge of the wound together because they are not packed tightly. And here, in this point, I turn off the damage, assuming that the cells have touched, and everybody goes back to the epithelial steady state. The E-cadherin goes back up, the EMT stuff goes back down, the cells reform the epithelium. Everybody is, again, happy in the tight epithelial state. This is the exact same simulation. However, here, I just knock out HHIP. So I set it to permanently 0, so it cannot pull back glee anymore. So what we see is the blue line here, glee goes up as soon as I turn on the damage, and it saturates at 1. And it turns on the rest of the EMT program. Remember this yellow module, which means that the whole system goes to a different state, which is the mesenchymal state. The E-cadherin, the glue, goes to 0. And this is when the cell completely detaches. And what's interesting here, that even after I turn off the damage signal, it stays there. It's a self-sustaining state. It will not go back into the epithelium. So the cell becomes sole. And this is like a simulation proof of this hypothesis I outlined here, showing that HHIP has this role in wound healing. We also validate this model. This is, again, a novel way of validating this model. We collected 46 different perturbation experiments from literature, and we use this network to reproduce those experiments and to see if they are actually reproduced correctly. And we have, if we just look at the direction, we have a 92% accuracy. So it's not just this behavior that we reproduce, but a lot of other behaviors. So as I said, simulations are cheap. That's why we do them. And they also show that we have some sort of understanding of the mechanism. However, the best way to make sure that we are right is to also conduct experiments. So, but this case, we conducted a targeted experiment, which means that we have grown a type of cell line, which is from alveoli. This is actually a cancer cell line, but it behaves similarly. The hedgehog pathway behaves in a correct way here. And we did what I showed in the video, that we grown the cell line and we scratched it. And we looked at the healing. And we also used PCR to measure some of these genes to see what happens if we knock down HHIP. So we had two groups of plates. On one set of plates, we had just control. In the other set of plates, we knocked down HHIP. And this is the statistics of a few genes. So blue is control, where we didn't manipulate. orange is where we knocked down HHIP. HHIP is lower. We expect that because we did that. However, we didn't manipulate anything else. We see CDH1, which is, remember, the gene that creates the glue, significantly lower in the HHIP knockdown. And one more I want to highlight, GLI2 is significantly higher in an HHIP knockdown, which confirms that the self-moderation hypothesis. So if I remove the self-moderating negative feedback loop through HHIP, then GLI goes up and it can turn on the EMT pathway, which then can cause problems. So the problem with this experiment is that we use a cell line. And cell lines are immortalized, they behave weird, biologists don't really like them, and it's also a cancer cell line, so we wanted to look for evidence somewhere else too. And in fact, I know this will be a difficult figure to explain, but I'll try my best. So we have single cell data from actual lungs of people with and without COPD. So the top part is from a data set that was generated from samples from our 70 people, out of which maybe 30 had COPD, the rest, some of them had other diseases and then controls. And from these samples, with single cell RNA-seq technology, every single cell's expression of RNA can be measured for every gene, which means that we have not just as many points as many people we had, but as many cells we collected, which can be hundreds of thousands of points. And we compared for the same genes, the measurements, as with the experiment before. The yellow is lung samples from COPD people, control is lung samples from controls. We see HHIP is lower in COPD, GLI2 is slightly higher, not a big difference, but the reason is, I'm not surprised by this, is that this happens pretty quickly when there's just an injury. Then GLI turns off back again. So on average, I don't expect GLI to be more activated, but even a slight activation is pretty good. We see also that the glue gene is lower in COPD. However, remember from the beginning of the talk that we started out with those SNPs. We started out with those mutations. Here we don't know anything about those mutations. Can we link those mutations to the mechanism? And this figure is from a data set that is fresh out of the oven generated. We have lung tissue samples from about 100 people, and one of our collaborators generated the single nucleus RNA-seq data, which is measuring the expression in the nucleus of every single cell from 100 people. And the interesting thing is that for these 100 people, we also have the full genome sequenced. So we know every letter of their DNA. So we can look how those single mutations are linked to the whole story I told here. And what I'm showing here, these RS and the number thing is just a SNP ID. So these are basically SNPs. This is the lead SNP for HHIP, which just means that the most significant mutation close to HHIP. And these are basically cell types in the lung. 81 and most importantly, 82 is a type of cell in the lung that is responsible for the healing. And the dots represent the activation of the EMT program. Remember this part. So if EMT activates, then the cells detach. So this is the p-value, the size of the dot is the p-value of the EMT, the whole pathway What is the significance of the whole pathway activating? So if a cell, regardless of COPD status, if a cell carries this mutation, there's a very high chance that the EMT pathway will be activated in these lung cell types. This is the story this figure tells, basically, that the most significant HHIP-linked mutations increase the likelihood of the EMT program activating and leading to aberrant wound healing in the lung. So to try to bring the whole picture together is that what we did here is that we started with Bob and Steve, and we were like, why is one guy susceptible to the disease and the other guy is not? And we compared their genomes, and we compared large cohorts of people, and we identified with statistical models the certain mutations. With the mutations, we can identify certain genes that are close to those mutations, but we still don't know what the genes do. And then we can construct these type of hypotheses using network models on what is actually the role of the gene in the wound healing. We can verify them multiple ways. First, using simulations. This is the in silico validation. Then we can validate it in vitro. And this is not exactly in vivo. We had also data from mice where HIP was knocked down. But this is supported in evidence in these large data sets. And why does this matter? If we understand the mechanisms by which these mutations lead to the disease, then designing therapies, designing interventions becomes much more easier because we are not shooting in the dark. We already know what we expect. And we can actually use these type of network models to test different drugs. We can we know what certain drugs manipulate, and we can play around with that. And we can even figure out ways on how to intervene. We can find targets on what kind of targets do I want to target with the drug, what kind of gene do I want to target with the drug to maybe prevent this from happening, or even potentially reverse a certain condition. So with that, I have one more figure. Remember that we don't have just one suspect. We have a lot of them. Right? We started with 80 something, we narrowed it down a little bit. So one mechanism will never explain the disease. So it's an important part. It's important to understand what HHIP does, but we have a lot more. And we have a separate project where we are looking for protein-protein interactions in lung cells. And we actually identified this little core network where we have 4 of these genes that have mutations associated with them, these risk SNPs. And all the other proteins in this core network are also shown to have higher or lower levels in COPD lungs. So if we want to understand the bigger picture even better, then this is something that we need to study. Now, you see that there are no directions on these arrows. We don't actually know what these interactions do. So that needs to study. What we know that these proteins stick together and they do something, but we don't really know what they do. So this is the next direction we want to go to. And I think I already gave you a summary, but basically we know that these diseases, and whatever I said here, as I said, can be generalized to other complex diseases. And today I just presented an application where we used network models and different types of experiments to actually try to understand a mechanism of how our genetic susceptibility works. And just to repeat, by understanding these mechanisms, we are better equipped to design therapies and maybe potentially improve the lives of other people. With that, I'd like to thank my collaborators. The leader of our lab, Ed Silverman, he is one of the most prominent figures in COPD research. He created one of the biggest cohorts. It's called COPD Gene. It has tens of thousands of people. I'm actually not sure about the figure. With smokers and we also have different time points. We follow up these people every 5 years. And this is one of the reasons we have this rich data to work with. And Erzsébet is also our collaborator, and Kimberly Glass is our network, my mentor in network science. So the paper with the results around HHIP is currently under second round of review at PNAS, but we have a link for bioRxiv if you are interested. And with that, thank you for your attention.
Thank you very much, David. It was a very interesting topic, and going through the crime scenes and identifying suspects, so it was really interesting. Now, if you have some questions, comments, we can discuss a bit. Yes?
One department at our institute, you know, at the Transylvanian Institute of Neuroscience, works on a topic like this. Actually, Bea is involved in a similar topic, so you really helped me understand a few things here. But I want to ask you that, so they are working on a similar study, but on neurodegenerative diseases.
Okay, yeah.
And so you use models here, so only the first part is similar, of course, because they do not use Boolean models.
Yeah.
But I want to ask you, you have models for epithelial cells, but do you have models for other types of cells too? Or no? Not yet?
Yes, but not for neural cells.
Erzsébet.
Yeah.
Erzsébet, I think she told me that she worked out some models for neurons because her students...
She had a student who was focusing on Alzheimer's, that I know. Yeah.
So maybe I should ask her more details.
Yes.
But I think it would be an interesting approach to combine so the way you continue the study using Boolean models, it would be very interesting because as I told you, the beginning is very similar to what they do, but you continue it in a different way with these interesting Boolean models, and it could be very interesting. So you do not have models for neurons yet, but it could have a great potential because it's really important.
Yeah, so I 100% agree. So Alzheimer's is number 7 there. And I have been actually thinking about that because in the case of Alzheimer's, it has to be some sort of a different mechanism, but it's also a sort of degeneration. So cells die in the brain for some unknown reason, and then the brain starts to shrink. And it has to be, again, some sort of an interplay between multiple factors. So I think we have this larger model, let me go back here, that has some core features. So here, every big dot is an entire module. It has multiple things in it. We just simplified it. This part here is very ancient. in an evolutionary sense. Also, the hedgehog pathway that I've been talking about, it's still in fruit flies. We just have different names from the genes, but it's there. I think that's where it was discovered. So here we have things like the cell cycle. Cells, like every single cell has the cell cycle. And this is a core control part of the cellular network. And the epithelial part, which is built around it. So this is the part that represents epithelial cells. These are the genes that separate epithelial cells from other cells. I think that the task, if you want to look into neural cells, is to take this core and just build around it the types of genes and the network of those genes that separate, that differentiate the epithelial cell from the neural cell. So I think it's doable, and I think your sister has already done some work on that.
Yes?
I have a technical question regarding your results on the Boolean simulations.
Yes.
Because you did some statistical averaging of several simulations, but if I do not understand, if you are starting from the same initial state,
Yes.
then why is the fluctuations, and shouldn't be all the simulations, shouldn't they lead to the same result if the initial state is the same?
That depends on the update type. So this is a very technical thing. So if we use synchronous update, then the system is deterministic. And then you're 100% correct that we will have the same result. There it's not interesting. This approach, that doesn't work. And depending on the system, we have to think about which type of update to use. So on synchronous update, we update every node at the same time based on the previous state. When we use, and in this case, we use something we call general asynchronous update, where we pick a random node and we update that. So every time step is a single node update that is picked randomly. So there's an inherent noise simulated here. So the system is not completely deterministic, but it can find something we call complex attractors. It's a subset of states that it can randomly jump around, but it is still noisy. And the probability at which it visits certain states is different. And by averaging these out, we can actually collapse these probabilities into like a single line. So there we simulate a certain inherent noise by adding this general asynchronous update. And this is why this happens. But that's a great question. Yeah.
I would also have a small question. So regarding your Boolean network model, you mentioned that the edges are based on previous studies, so
Yes.
on empirical observations. And in this case, are we sure that the missing edges are truly missing, or?
You touch on a great point. We are not. And that is something that we have to face. I have to also mention another limitation, that these models are completely epigenetics agnostic. Epigenetics are not considered here. Because epigenetics, what would it mean? It would mean that the regulation is also noisy, because the DNA can be packed up in these so-called histones and it's not accessible. There's another mechanism called methylation that makes it difficult for genes to get activated. So that adds another layer of noise. What you're asking, that the missing edges, we basically have to work with the assumption that what we have is enough to explain the phenomena that we see. And sometimes it's not, and that's why we conduct these other type of studies where we are actually actively looking for edges. But we are dealing with a lot of noise, and that's why I would say this is a somewhat conservative approach. And by conservative, I mean that I only include edges that we have pretty good evidence for. So it's like I only work with the stuff that we have like solid evidence for, and whatever is missing might come up later, but in this case it actually worked out pretty well. But that's a great point. We always have a paragraph about this in the papers because...
Could your simulations provide a feedback for the biologists to focus on 2 genes? So you may have noticed in your simulation that there may be something there, or the results would be better, and then to tell the biologists.
Absolutely. Actually, I mean, just to give a history of this, like we discovered this mechanism with playing around with the network idea, and then that's how we told the biologists to try this experiment and measure these specific genes, because the simulation gave us the idea that this is the direction we should go in. So absolutely, the simulation is basically almost, you can think about as like a video game where you can just play around with genes and see what happens.
Okay, thank you. I have verified the YouTube channel and there are no questions, so I think we can close our today's session. So thank you very much, David, for this enlightening presentation. It has a privilege to see how the rigorous models of physics, let's say the roots you have planted here at the Babes-Bolyai University, are now bearing fruit in the fight against complex diseases. So thank you very much for coming inclusion. Thank you for having this presentation to us. As this session draws to close and we approach to winter break, on behalf of the STAR-UBB Institute and the entire Studium Generale team, I would like to wish you all a peaceful holiday season. May you have Merry Christmas and a Happy New Year filled with health, inspiration and new discoveries. Thank you and see you in 2026.